In this video, I'm going to introduce you to the role of DNA polymerase and ligase, and we're going to talk a little bit about what we call the lagging and leading strands of DNA as well. And these we all see during the process of replication. Before we get into the detail of how DNA polymerase works together with primase and with ligase and helicase, we're going to talk a little bit about the specifics of the structure again here of DNA and talk specifically about the reaction that's catalyzed by DNA polymerase. So remember that in DNA, and we've talked a little bit about this before, those 5' ends are always going to have a free phosphate on them, and then the 3' ends, like this one down here, are going to have a free hydroxyl group, and so this would be the 5' and 3' ends of this upper fragment of DNA here. And then you can see in the middle here I've drawn out those base pairs, so remember GCs have 3 hydrogen bonds holding them together. ATs, on the other hand, like this one here, have 2 hydrogen bonds holding them together. Now, one of the things about DNA polymerase is it catalyzes DNA in a 5' to 3' direction, meaning it can only add nucleotides to the hydroxyl groups that are at the end of a growing chain. And so what it's going to do in particular is it's going to take a triphosphate, so something like adenosine triphosphate. Let me go ahead and draw it out here for you. So there's one. Whoops. There's our second phosphate. And here's our third phosphate. And again, this is attached to what we call that 5' carbon, which is right here. So here's our oxygen. And then this would have an OH hanging off of it at the end here, like so. And let's say that this time we're going to add an A. So A is our nitrogenous base, or adenine. Well, what's going to happen here is this enzyme is really catalyzing a dehydration reaction. And the way that it's going to is it's going to use energy from these last 2 phosphates on this triphosphate. And so what's going to happen if we kind of draw this out is these 2 phosphates break away in what we call a pyrophosphate, or sometimes it's labeled as PP with a little I for inorganic phosphate. And that pyrophosphate provides the energy for this reaction. And so what's going to happen here is as those pyrophosphates leave, that releases a huge amount of energy, which is going to allow for this phosphate right here to bond to this carbon right here. And it's going to lead to the loss of water in the process. And so at the end of this, and I'm going to erase a little bit of this here at this point just to kind of show you what's occurring. Whoops, that's not an eraser. Here we go. So those 2 phosphates are burned. This OH goes away as well. And ultimately what's going to be created is a bond here between this carbon and this particular phosphate as well. And again, we're losing water in the process because this is a dehydration reaction. All right, so let's talk a little bit about the detail, how DNA polymerase actually catalyzes the reaction and how it looks now when we take into account this whole replication fork. But before we do that, let's make sure to label a couple of things. Here's our helicase again. This is the enzyme that is moving in the direction of double-stranded DNA to unwind it. I'm going to go ahead and label my 3' and 5' ends of each DNA strand, as so. And then in green again are our primers, and these were created by the enzyme primase. Now what DNA polymerase is going to do is it's going to latch on to this double-stranded nucleic acid that is created partially of the RNA primer and partially of this template DNA, and DNA polymerase then is going to move in this 5 to 3 direction to put down the complementary sequence to the template DNA. So for example, if our template has the sequence from 5' to 3' TAC, then that DNA polymerase, is going to put down the opposite sequence, GTA, again in a 5' to 3' direction as shown here. And as this DNA polymerase is creating this strand of DNA, so let's just kind of fast forward it to this point right here, what it's going to start doing now is when it encounters these single-stranded binding proteins, it's basically going to bump each of those off of this DNA at this point as this DNA's polymerase continues to move in that 5 to 3 direction. This is going to happen on both strands of DNA, and so we could kind of model the same process on the top strand where DNA polymerase binds and then is synthesizing this new DNA here, and again, as it's moving in that 5 to 3 direction of the new strand that's being made, that DNA polymerase is knocking these single-stranded binding proteins off of that DNA. Okay, so here is a replication bubble, as we've talked about. What we would have had is, if we go ahead and take a look at this again, we would have had a primer associate probably right about here and right about here, and DNA would be synthesized in the 5' to 3' direction of that strand being made. And what I'm drawing here in purple from that green primer is what's known as the leading strand of DNA. The leading strand is going to be the strand of newly synthesized DNA that follows directly behind the helicase at the replication fork. And so these particular strands of DNA are being made in the direction of those replication forks. Now, what you can imagine is that as these replication bubbles increase in size, it's going to expose some single-stranded DNA behind the primer that has not yet been replicated, and this process will continue. And so what's going to happen in these cases is new primers are going to have to be created, and as those new primers are created, we get new synthesized DNA, and this time I'm going to show these in kind of a bluish color here and here. And then as that DNA replication bubble again increases in size, new primers need to be synthesized, and we get another short fragment made. And these short fragments are what's known as the lagging strands. These ones are shorter fragments, usually maybe 200 to 300 nucleotides in length, and these are ones that are discontinuous as opposed to the continuity of the leading strand, and they're created as that replication bubble opens up. And what this creates for us then are a series of short fragments that together are known as Okazaki fragments. And these are named after the scientist who first discovered them while studying this process of DNA replication. And so all of this replication then takes us to this last step in the process. And really there's going to be a couple of things that we have to address here. First, these primers, don't forget, these primers are made out of RNA. And so somehow they have to be removed. And we'll talk about that. The second thing is that in the DNA, from the primer to the next one, we're going to have little gaps that exist in the sugar phosphate backbone. And so we have to ask, how do those get fixed? And so we're going to talk about those two steps here. So I'm going to erase these real quick. And what I'm going to talk about is the role of DNA polymerase in finishing off this replication process. And we're going to draw DNA polymerase at one of these right here. And recall, DNA polymerase, in order to work, needs not only a primer, but also in double-stranded DNA, but it also needs a template to read. Well, one of the cool things about DNA polymerase is it has the ability to remove the RNA primer, like this one right here. And so as that DNA polymerase proceeds in this forward direction, what we're actually going to find is it's going to replace that RNA primer with DNA. And that would occur up here as well. here as well. And so all of those RNA primers are going to be replaced by RNA polymerase going to be replaced with DNA by the DNA polymerase enzyme. But that doesn't address all of these nicks that we've got in the DNA. For example, I'm showing one up here at the top and another one here at the bottom. And these nicks are basically a break in the sugar phosphate backbone that make up the DNA. In order to bond these last sugars and phosphates together to create one continuous strand of DNA, our cells employ the help of an enzyme that is known as ligase, or DNA ligase. And what DNA ligase does, if we go to where those two arrows once were, DNA ligase basically acts like glue, but it's going to catalyze the last dehydration reaction between a sugar and a phosphate. And so DNA ligase is going to come in, we'll draw it like a little trapezoid, and as it's coming in here, it's going to help to fix that sugar phosphate backbone, basically pasting these two strands together to now create a continuous strand of DNA at this point. So I know that this lecture has become a little bit longer than what I anticipated, but I do want to just kind of readdress the role of the different proteins that are involved in this process of making DNA. We first talked about helicase, which is the enzyme that unwinds strands of DNA. We talked about the role of primase in creating the RNA primer, and that RNA primer is important because it's the beginning site for DNA polymerase. We did talk about DNA polymerase and how it's involved in making new DNA in a 5' to 3' direction. Of course, it's making a strand that's complementary to the template. And then we talked about the role of single-stranded binding proteins and keeping those two template strands of DNA separate from one another until DNA polymerase can catalyze the reaction. And finally, we talked about the enzyme ligase, which helps to paste together or help to create a bond between sugars and phosphates between nucleotides that was not created by DNA polymerase.